extinction trends and weigh the pros and cons of bringing extinct species back to life, and are made possible with support from the University of Oregon's Wayne Morse Center for Law and Politics. During this evening's program, you can use the Q&A or chat features on Zoom and Facebook to reflect on the talk or add your thoughts, to engage with other viewers, and to ask questions of our speaker. I'll be collecting your questions across both platforms, and we'll get to as many as we can at the end. Also, we'll be providing a link to a brief survey in the chat box on both Zoom and Facebook. I'd appreciate it if you could take a minute or two to fill it out, as your feedback will help us improve future virtual programs. Before we get started, I have an upcoming program I'd like to share with you. Next week, on Wednesday, March 3rd, our monthly Ideas on Tap program is back with UO sociologist Claire Herbert. Claire will join us for a discussion about Eugene's affordable housing crisis and what it means for the city's students, former prisoners, and others at risk for experiencing homelessness. If you can't make it to the live program, we post recordings of all of our virtual talks to the museum's YouTube page on the following day, so be sure to check out any talks that you've missed. Finally, if you're able, we ask that you please consider making a secure, tax-deductible donation of $5 to $10 at the web address listed on the screen to help keep museum programs like this one accessible to all. That's giving.uoregon.edu/mnch-gift. Your donation directly supports the museum's educational programming, bringing science and culture adventures to Oregonians of every age and in every corner of the state. Thank you. And now it's my pleasure to introduce our speaker for this evening, Dr. Ross MacPhee. Ross is the curator of mammals at the American Museum of Natural History, as well as a professor emeritus at the Richard Gilder Graduate School. His research interests include recent mammalian extinctions, concentrating on the loss of megafauna during the end of the Pleistocene. His research seeks to clarify the causal patterns behind these losses, most recently by studying population dynamics of fossil species using ancient DNA methods. Welcome, Ross.
Thank you. It's a pleasure to be here. Can you see my screen?
Yes, we can.
Good. It's always the same with these Zoominars, you never know. Well, I want to thank everyone, even though I can't see any of you, for coming out tonight to hear about de-extinction, which is certainly one of the most interesting and provocative issues in modern conservation biology. And I'm not advancing. What is that? There we go. So what is this thing called de-extinction, and why should you care? In one sense, it means exactly what the word says. Restore to abundance populations of extinct species and help them to prosper. The lines are a bit blurry between completely extinct and just dreadfully endangered. I intend to discuss examples of both, but my main emphasis will be on the completely extinct and whether they can or should be brought back. Realizing de-extinction, making it happen, will require the application of techniques that could scarcely have been imagined even a generation ago. In short, this talk is not going to be your dad's paleontology. We're going to be talking about a lot of things that involve modern biology, as well as the old biology of physical remains, fossils, of organisms that died a long time ago. I want to emphasize that there's a lot of interest in de-extinction under different names, of course, but the idea of bringing things back or bringing them back from the brink. A lot of these organizations are very well known for their conservation activity and are increasingly moving into the kinds of genetically mediated techniques that I'm going to be talking about today in order to do our very best for the organisms that are still with us. I'm also going to be briefly talking about bioethical considerations at the very end of the talk, I would like that, in fact, to be where I get a lot of questions, because it is important not only to think about bringing things back, but what are you going to do with them once they're here? How are you going to take care of them? Things of that nature. You'll see this slide again at the end of my talk. Okay, let's get started. The image in front of you is an effort to show the diversity of organisms that lived around the famous La Brea locality in Southern California, in Los Angeles, about 11,000 years ago. You can see it's just filled with large creatures. But all of them, all of the ones that you see here, have only one thing in common, which is that they are all dead and gone. So all of this magnificent wildlife, the giant ground sloths, the zebra-like horses, the saber-tooths, giant bison, mammoths, pronghorns, on and on, are no longer with us. So the first question that arises, why wouldn't we, if we could, bring at least some of these critters back? Wouldn't that be a great achievement? Wouldn't that be something well worth doing? So let's consider a few arguments in favor of this. I'm going to have a little bit of fun with them as well, because this is a very serious topic. It's good for a little levity over the bad bits. Okay, one reason for de-extinction, it's because it's our fault that so many have disappeared. This is obviously a major concern in conservation biology today. And certainly, many paleontologists believe that the megabeasts I just showed you disappeared at least in part because of human depredations. So the idea would be, okay, our bad, let's make it up by reinventing some of the disappeared. Restorative justice, making amends. Here's another idea. Yes, there are a lot of endangered species today, some of them very close to ending their tenure on the planet. The argument here would be that we should try to bring back the already disappeared, because we're going to learn so much about what we can do. So we'll have an arsenal of new ways to help species that are in bad shape, that are still with us. If that still doesn't do it for you, then how about this? Wouldn't it be way cool to see a real live mammoth? I mean, seriously, who among us wouldn't spend a few bucks just for the opportunity to see this maybe a few years from now? You get the picture. So let's ask the next big question. How might de-extinction actually be accomplished? There's several ways, depending on what you are willing to call de-extinction, what you regard as a success, and whether bringing a taxon back will really lead to anything or just be a 9 days wonder, are issues that we have to confront. I'm going to be talking about approaches to de-extinction by reference to three main types. Selective breeding, in this case, in an effort to reverse or to recover the primitive or wild type. Reproductive cloning, using somatic or stem cells for that purpose. And then genomic engineering, using CRISPR and other methodologies that I'll be telling you about as we go along. So we can actually go in, change gene sequences, and do apparently wonderful things. Okay, selective breeding. This is by far the oldest and simplest method of doing anything in respect of de-extinction. Humans learned relatively early that they could manipulate nature in numerous ways. Controlling the breeding of desirable plants and animals was one such way, a very important way. This is what humans do. We mess with stuff. During the course of our history, we have conducted thousands, probably tens of thousands of experiments in individual domestication to get what we wanted. We want bananas that go yellow without immediately rotting, bonsai trees, and chickens that seem mostly to consist of breast meat. All of these are interventions by selective breeders to get desirable results. Now, with de-extinction, we're actually reversing the process. We want to go back in time, like we had a time machine. We want to go after the characteristics that made the ancestors in the same lineage as the animals that are still with us, to get their appearance at least back so that we can qualify that as an example of de-extinction. And the one I'm going to briefly address here is the aurochs. The aurochs, Bos primigenius, is a species or a near species. It's a member of the cattle family, closely related to our domestic cattle, that is thought to have died out finally during the 17th century in Central Europe. Now, as it so happens, breeders, rewilders in particular in Europe, have been very interested in trying to recover the aurochs, at least phenotypically, its external appearance. And what they do are crosses, and this is probably biology 101 for all of you. You go after the characteristics that you want by breeding animals that share them. And you find over time, and hopefully, that the successive generations look more and more like what you want and become a pure breeding line, at least for those traits of interest. And in the case of the aurochs, what we've got are animals that now really do look like those old cave paintings. The horns are right, the color is right, the size is right, all of that. But the point is that that's all you've got. You've got the external phenotype. That's what you were selecting for. That's what you end up with. Whether the modern looky-likey aurochs is anything like the one that lived in the past is open to question. Behaviorally, physiologically, resistance to disease, these are things we'll never know. So conclusion here for artificial selection is that as a method of de-extinction, of bringing back the dead, offers little. The results are superficial, even if for other reasons you like these results aesthetically. Reproductive cloning. This is a big subject. You're only going to hear a small bit of what you really need to know about this kind of methodology. We're going to go straight to the first successful example of a vertebrate being cloned from an adult somatic cell. This is the famous Dolly the sheep. This is her dad, Ian Wilmot, who was the scientist in charge of her development, so her creation. Basically, Dolly had three parents. She had one, which we're calling the cytoplasmic donor, who produced an egg for this purpose. She had a nuclear donor, the mammary cells of which released nuclei that were then implanted in the cytoplasmic donor's egg after its own nucleus had been removed, as this animation will show you. There goes the nucleus, it's thrown away. What happens next is that the nuclear donor gives over an example of the nucleus that is implanted in the cytoplasm, in the actual egg cell, which is now just cytoplasm. And the next stage is to zap it with electricity. You can also use various chemical agents that do a weird thing. They turn this cell back into its primitive state. So it's almost like a stem cell. And it begins to spontaneously mitotically divide, just like it would if it was a real embryo or an embryo gotten in the usual manner. After a few divisions, what happens is that the blastocyst is taken, implanted in the surrogate, the third mother, and presto, within 5 months, you get Dolly. I might add after about 300 tries before they were successful the first time. So what is to be said about this? This experiment demonstrated that it was possible to take a fully differentiated adult somatic cell from any part of the body, wouldn't have to be necessarily a mammary cell, and effectively reprogram it so that it became a functional zygote when introduced into the necessary cytoplasm of the egg, and grow into an entirely new organism. Now, you can imagine, those of you who are my age can even remember, back in the 90s, when Dolly was first announced, this was a huge thing. It was also a huge thing for people who were thinking at the same time about, well, if you can do this with any mammal, what about a mammal that's long been dead? If there's examples of it that are very superbly preserved. You may remember seeing images like this of one such sample, the Zharkov Mammoth from northern Russia, in the Taimyr Peninsula. We're not seeing much of the specimen because it's still encased in frozen mud that was cut out of the tundra with its tusks attractively attached to the front of it for PR purposes. And then it was shipped off by helicopter, as you can see in the second image, over a long distance, taken to a lab where it was supposedly going to be cloned. This was the beginning of the frenzy over cloning the mammoth. Now, how would this have taken place in terms of what was understood at the time, at least by the people who were making the argument they could do it? Well, what you need is a viable cell, an intact nucleus. Otherwise, it's a no-hoper. Cloning is not going to go anywhere. So the idea was that you would get a pluripotential stem cell from an Asian elephant, which is the closest relative of the woolly mammoth, which is an important thing. And you do much the same kind of operation as you saw with Dolly. By nuclear transfer, you would put it into an enucleated egg coming from a female Asian elephant, zap it, and then put the zygote into a female who would then give, one hopes, live birth. This first generation should look a lot like a mammoth. There's only very few genes that are in the cytoplasm of an egg, and everything else should be under the control of the nucleus that was introduced into that cytoplasm. But here you get the first problem, which is that a single female is only going to produce another calf on about a 2-year basis, because it takes almost that long for a pregnancy to occur. So over time, here you've got to put your mind in that of somebody who wants to do this operation. You're probably going to have to cross at least some of your mammothy looking individuals back with a female Asian elephant. You'll get a hybrid, and all you can do over time is to keep going for the individuals who look most like mammoth, keep breeding them. You'll get a sport once every so often. This is what always happens with pure breeding lines. It won't look very much like a mammoth, but you exclude it from further breeding. Okay, doesn't that sound wonderful? This was the dream. And here's how reality bit back. I don't know what you know about mammoth mummies, but here's what I know, having collected a few in my time. They're like road pizzas. They've been through it all. They don't just get deposited, covered with dirt, and stay flash frozen, preserved until some paleontologist comes along and digs them up. They look like this one. In fact, this is a very well-preserved skull head of a woolly mammoth. It's not the Zharkov mammoth, because its head was in terrible shape, but it gives you a sense of what even the best look like. So here's the problem. Everything about this is deteriorated. The DNA has undergone damage and breakdown, which is what happens with the DNA molecule as soon as there's no energy coming into the system anymore after death. And there were no, as you might imagine, there were no viable cells in the Zharkov mammoth. It had been in the ground for about 14,000 years. Under what dream state could you possibly imagine that there would be anything viable? So that was it. That was the brief chronicle of the Zharkov Mammoth, the cloning the Mammoth frenzy, its hype, it was all over. Having said all of this, I don't want to give the impression that cloning is a useless kind of methodology for assisting particularly critically endangered species. Revive and Restore, which is one of the organizations that's involved in this kind of work, just last week announced the birth of a clone of a black-footed ferret. Black-footed ferrets are almost extinct. And this was the first time that a clone had been successful using a frozen egg from a female that had died some years before. And here we have Elizabeth Ann, the first cloned black-footed ferret. So great success. Another possibility of doing much the same sort of thing would be with the northern white rhino subspecies. You may have heard that the last wild living rhino, Sudan, died a few years ago. There aren't any more in the wild. There's a few in zoos, mostly aged females, but there's also frozen zygotes. And the idea is that maybe this is the way forward. We try and do exactly what I've been talking about methodologically with crossing the nucleus from one of these preserved stem cells or gametes of the northern white rhino, and using an egg from the other subspecies, the southern white rhino, you see whether or not you can actually get a positive result, which would be another cloned animal based on an individual who had died long before. So it's not quite de-extinction, but it's very close to it. And it's a very important undertaking. Our next and last example is genomic engineering. Now, genomic engineering is a really interesting field, and I wish I knew a lot more about it than I'm able to pass on here. I'm only going to concentrate on the basics just to give you a scintilla of an idea about what it might be able to do for the kinds of questions we're asking here. The first thing about synthetic biology that I want you to remember is that it's actually engineering in a way that cloning is not. With cloning, you're just basically working blindly. You're hoping for the best. You've got what you hope is a viable embryo, you zap it into mitotic division, you transplant it into a surrogate, and then you wait and you hope. Synthetic biology, with respect to what we're talking about now, is different in that it holds in principle that it's possible to do almost anything with the genome, including building organisms and functions that have never existed in nature, at least in the species that one might be talking about. Here's an example. These are mice. Two on either end have had a gene transmitted or transgenically placed in their germlines from a fish, I believe. And what this means is that under blue light, in parts of these animals where this particular protein is expressed, they fluoresce. Yeah, kind of weird, I know, but this can be done without affecting other functions. It's only one gene. And it doesn't seem to bother the mouse very much, since its only effect is seen under blue light. Larger introductions of foreign DNA, however, are a different kind of thing. That may result in damaging the new host's developmental systems, with the result that you get an abortus, not a newborn. So you have to be careful. What it all means for de-extinction is that we can't build a complex species from scratch, like a bird or a mammal at this point, and we may never be able to do that. What we need is a so-called scaffolding. What this means is the known genome of a living species that you can utilize to determine how and where it differs from some relative extinct that you're interested in reconstituting. So our case here is Asian elephant in the upper left and the woolly mammoth. Although they're put into separate species, even separate genera, biologically they're very similar. Their genetic inheritance is about as similar as ours is to the chimpanzee or bonobo, which is over 95%, even up to 99% for protein sequences. So what this means is that you can use an Asian elephant to provide scaffolding because it's going to be extremely similar to the genetic code of the woolly mammoth. And you can go through it and find places where it differs. These would be the traits that are unique to the mammoth. So the theory here is that you need a way to go to those sequences and change them, replace them with the kind of sequence that the mammoth had. All of the other steps are downstream, the zygote, making the embryo, doing all the rest of it, but that's the start. Now, this seemed like so great an opportunity that George Church, seen here uncharacteristically out of his lab at Harvard and looking for bones like I do, he thought that, yeah, we can do this, or we can at least make a very spirited effort to try and reconstitute from an Asian elephant scaffolding what the genome of a woolly mammoth should look like. So, an astounding thing. Now, the amazing tool that's needed to do this is called CRISPR-Cas9. In fact, there's many kinds of CRISPR methodologies, but I'm going to talk about the original one that is still used for many purposes, called Cas9. Let's look at this cartoon to sort out what it's good for. And we're looking at the series of images that's in the middle of this diagram, standard CRISPR-Cas9. You can think of this, this entity, CRISPR-Cas9, as being basically a drone. A drone flies around autonomously looking for a target, looking for something that's programmed to do, right? And in this case, what you've got for the CRISPR part, which is in blue in this particular diagram, it's going to fly through the strands of DNA in the cell, looking for a very specific sequence that mirror matches a small bit of sequence that's attached to it by engineering of RNA. This is in blue. So what you have to imagine is that this molecule is going to be moving along step by step across a chromosome, looking for this particular sequence that you're interested in targeting. When it finds it, it gloms onto it, at least the Cas9 part of it does. This is an enzyme that can cut DNA. This is its role. So it attaches to the identified sequence and works like a pair of scissors to cut the DNA at the chosen spot. Now, this is incredibly interesting. There are questions about how accurate CRISPR and Cas9 can ever be. It's being worked on continually. And all I'm going to say on that particular point is that it's certainly more accurate than any other method we have using bacteria or anything else. The idea also is that with the cut having taken place, you can do things. You can introduce, for example, DNA that you place there using something else like a bacterial vector and slot it in to the cut area. And I think you know where this is going. If you have a situation where you've got a gene for which the Asian elephant and the mammoth differ, then using CRISPR-Cas9, you go in, make cuts at the right places, and if necessary, replace what you've cut out with Asian elephant sequence. You've done something amazing. You have created a gene that hasn't existed for 10,000 years. Now, What are you going to get? That's part of the problem. Let's begin with the idea of how many engineered replacements of genes would you have to do. It's very hard to predict at this point. But surely, only a handful would even be feasible, considering the time and expense that would be involved in all of this. There's 3 billion base pairs making up the genome of the average mammal, so you can see the magnitude of the task. And it wouldn't be necessary, perhaps, to go through the entire genome with this kind of methodology to achieve exact matches for every gene that's different. Because once again, isn't it partly all about appearance? If you have an animal that has the long hair, the fat deposits in the right places, short ears, the curvy tusks, then only a nerd like me would say, well, that's not really a mammoth, that's just an invention. And the point here would be that if you're trying through restorative justice, this is the other part of this, to recreate something that we might have had some degree as humans responsibility in causing the extinction of, then wouldn't this be, in a way, a wonderful thing? Wouldn't it tell us that humans can still play the kind of positive role that we must continue to play, should play, in the world as it is today? Okay, I'm going to now finish off with some bioethical questions, beginning with this one. Is it even feasible, ethically, as well as practically, to bring back the disappeared? And what do we really mean by bringing them back? One of the things that we surely need to think about is where we're going to keep them. If you're thinking, yeah, ground sloths, good idea. Tapirs, which used to live in North America, great. Cheetahs, which used to live in North America as well. All right. Well, one won't do it. One is the 9-day wonder problem. What you want is a population that is sufficiently large to be self-sustaining. And no population is going to be self-sustaining unless it's in a place where it can be sustained. So we need to think about this. If we're going to go into the job of recreating the mammoth, then where are we going to keep those herds? Under what circumstances are we going to keep them? And what will their lives really be like? You recall just last week or the week before, Texas had a brutal, brutal winter storm, the effects of which are still continuing. Not only did it cover the landscape, it also meant no food and often death for domestic animals that were left out because they couldn't be brought back to stables or couldn't be brought back to barns. So just think at this point. You spend great treasure in creating a viable herd of mammoths. It's walking the plains of Nebraska. You get a storm like this. You have no way of supplying quickly enough. So you get possibly a wipeout. So it's all come to naught. You need to think in terms of what kinds of species we really want to bring back. If it's just a matter of showing how good faith, showing the good faith we have, then why not go after the kinds of animals that have mostly died out in the last several thousand years? They're not panda-like, they're not mammoth-like, they're rodents and bats. That's where most species extinctions have taken place. Yes, I know, those are not the kinds of species that most people would consider worth the effort to bring back, but the point is different. The point is that de-extinction should have in mind bringing back species that can fulfill a useful role for themselves, of course. But another one that I think is very important is to stand as an example for what the future can be like on this planet, where we give over increasingly large areas for rewilding, for taking care of species that are in trouble, of recognizing that we above all, and perhaps only us, can make any kind of difference. The final point I think I want to make is just my fundamental question about de-extinction in the first place. I've been a critic. I'm not in favor of it for all the kinds of reasons that I've just brought up here. But here's one. Can the de-extincted ever really be treated as natural? And the whole basis for their existence now would be so unnatural. They wouldn't be here. They wouldn't be thought of unless it were for humans and human ingenuity. Is it not better, in light of what could happen, not only to animals de-extincted, but all the other problems that might be introduced by virtue of having megafauna in numbers back in a place like the modern United States? Wouldn't it be better to look after what we still have? Thank you.
Thank you so much, Ross. So we actually have about 20 minutes for questions. So I do want to encourage people, if you haven't already, to drop those into either the Q&A or the chat feature here if you're joining us on Zoom, or you can use the comments in Facebook, and I'll be collecting those as well. So we actually had one come in already, and it kind of ties on what you were talking about right there at the end. So an extinct species has lost its niche to surviving species as evolution operates. What niche will a de-extinct species occupy, and what current species could be displaced? So you can pick whatever example you'd like to illustrate that one.
A very good question. It's one of the things that one should be thinking about when thinking about the purpose of de-extinction. Niches are basically theoretical in the sense that they don't exist apart from the animals that are exploiting them, as it were. And it's not beyond question that if you, say, reintroduced mammoths into North America, that they would be displacing anybody else, if only because there are so few other megafauna herbivores like mammoths around today. But we can think of other cases in which it might be a very bad thing, and these might not be mammalian examples, they could be either invertebrates or other kinds of vertebrates, where there's already such species diversity in small areas that to introduce an outsider, which is what we're talking about, there'd be competition for resources. And there could be other things. There could be disease brought in from the outside that would affect either the de-extincted species or the ones that it was kind of levering aside so that it could have its own part of whatever the situation it was found in. I think in general, if de-extinction ever takes off in any guise, it's going to be restricted. It's probably not going to be about mammoths, I think, because for fairly obvious reasons, where to put them is going to be a real challenge. And it may be the kinds of animals that we would not even regularly think about. There's lots of opportunity in Australia, for example, for recently extinct marsupials to be reintroduced, if it were possible, to find the tissues, to do all the rest of it that would be required to make it happen.
Thank you. So we're going to switch back a little bit away from the ethical considerations that we have here on the screen and touch on something that you mentioned towards the beginning of your presentation. We have someone asking, there appears to be a biological barrier to successful live births from surrogate cattle after being implanted with engineered auroch embryos. Any idea why this happens?
I've heard that, and I don't know the functional basis for it, but it is the kind of thing that happens with hybrids, which is in effect what we're talking about, all the time. And the reason in general is easy to understand. They're not the same. There's going to be differences genetically, which are going to affect various kinds of developmental pathways. And if those get interrupted or destroyed in some way, you're not going to get development. It's going to end. So it's by no means an easy thing to talk about cloning or talk about creating hybrids that are based on two different species. And this is another thing that we really have to keep in mind. This is animal welfare we're talking about now. What right do we have to play God in this kind of way when we're not omniscient? We're going to make mistakes. And there'd better be a good reason for making those mistakes.
So one of the questions that you have here on the screen ties into some of the presentation from last week, and we actually did have a question or two come in related to that last time. So I'm hoping you could touch on the question, should public funding be used to revive an extinct species?
Yes, an interesting question. Well, there's a question before that question, I think, which is, what are we trying to do in this country, say, with regard to these kinds of natural resources? Here, I'm not talking about extinct animals, I'm just talking about natural resources in general. If we're of a mind to preserve them because they're important unto themselves, then of course public funding is required. That's why we have a national park system. It's why states have their own park systems. It's because it's considered a benefit of being a citizen of this country to enjoy nature, wildlife in this kind of way. So with de-extinction, if we're talking about bringing back a specific animal, if there's a benefit that extends, this was one way to look at it, I don't necessarily endorse it, but if there's a benefit that extends more broadly than just to the animal itself, because it does a job. For example, it keeps grasslands so that they're not invaded by forest species. There's nobody doing that now except cattle and horses themselves are not permitted in a lot of areas. If that was seen as a benefit, then okay. But all the rest still applies. What are their lives going to be like? Who's taking care of them? Who has taken on the obligation in perpetuity of looking after animals that have been created in this way? In effect, they're humanity's children in a real sense. They wouldn't exist if it weren't for us.
So we've just had a question come in that kind of relates back to this point that you just made of why would we do this, as being that first question that we should ask. And someone is wondering, isn't there a Russian, I'm guessing, researcher trying to bring back or emulate the effect of mammoth herds on the tundra in order to combat global warming?
Yes, there is. Sergei Zimov. For this talk, I had thought that would be an interesting example to talk about, so I'm very pleased I can do it now in the form of an answer to your question. Sergei Zimov is a very well-respected, very well-known ecologist. And for the past 20 years or more, he and now his son have had the idea that it would be possible to reconstitute what's known as the mammoth steppe by introducing the kinds of living megafauna we still have. Now, first, what is the mammoth steppe? The mammoth steppe was a grassland that remarkably, in the last ice age, existed through the northern part of Eurasia, all the way over to North America, to Alaska and Yukon, because the Bering Land Bridge at that time was above water. So you had this largest biome on the planet back in the day, in the late Pleistocene. Now, this was interesting for at least two reasons. One, this is still very far north. So what's a grassland doing there now where there's only wet tundra? And secondly, how is it maintained? Zimov argues it was maintained by the animals themselves, that the megafauna were so-called keystone species, who in their millions, which is what you would need to do this, were able to not only keep the grasses cropped and the trees down and all of that sort of thing, but by manuring soils, turning them over, like kicking them, and walking along them and so on, you created an extremely rich environment that can nowadays be in fact seen and matched in the savannas of East Africa, where you do have a lot of existing megafauna doing exactly these kinds of keystone jobs. More than that, by virtue of keeping the grassland to grassland, you would provide opportunities for smaller species that wouldn't be able to make it in dense boreal forest as exists today or in wet tundra. So he started, he has a project in Kolyma, in Chukotka, in the extreme eastern part of Siberia or the Russian Far East. He's got horses, Yakutian horses, that are adapted to this kind of biome. He's got muskox, he's got bison. He would love to have mammoths, because mammoths are the keystone of the keystone. And he and George Church actually discussed the possibility of engineering mammoths, de-extincted mammoths, in such a way that they could stand the cold in northern Siberia and do their job of eating, manuring, turning over the soil and so forth. My objection to that is the same objection that I brought up with my Texas example. We're living in a period of radical climate change. We don't know what it's going to be like in future. Yes, it's getting warmer, but it's also getting more chaotic in respect of weather systems. And it would be a crime to go to the effort to de-extinct a mammoth, create a hybrid that in some way was part this, part that, so that it can survive up north, and then find that it doesn't make it, for any number of reasons. I don't think the mammoth steppe is likely, not because there's an ecological defect in Zimov's thinking. I'm not saying that. What I'm saying instead is that I see no prospect for having the literally billions of individual animals you'd have to have in order to keep the mammoth steppe, if it got reconstituted, a steppe and not revert back to tundra or to something else. Thanks for that question.
So this one kind of is great timing, building off of what you were just saying. Can you envision a scenario where we might be able to bring back an extinct species in a way that it could be done safely without major repercussions to the Earth's biosphere?
Well, I'm as interested as any of you. The kids out there I know have big eyes for all of this kind of thing. Wouldn't it be great? I just don't know what that would be at this moment. One way to think about all of this, in fact, with regard to de-extinction, is that the only successful examples of bringing back something extinct in the very narrow way that I've been talking about it are viruses. Specifically the Spanish flu virus, the 1918 virus that killed so many people. That was reconstituted on the basis of material that was extracted from burials in Alaska back in the late 90s. Now, I know you're not talking about that as something that you'd want to reintroduce, but it underlines the point how difficult it is to embark on this kind of thing with all kinds of repercussions that are also very hard to gauge what the effect is going to be. I mentioned previously that in order to garner support for any program of this sort, what you probably want is to show that it's not just wild-eyed academics' idea of a good thing, but that it actually confers a benefit to have some de-extincted species on the landscape. And the likeliest examples of that really are not megafauna. Megafauna are too big, too problematic to put anywhere where they are not already, and I wouldn't advocate that. So we're talking about something very much smaller. Let me give you an example that some of the people involved think a lot of as something that's worth doing. This is again a revive and restore undertaking. I urge you to go to their website. This is the premier organization that's involved in this kind of work. And in addition to their interest in de-extincting the mammoth, they're interested in de-extincting the passenger pigeon. Passenger pigeon, what is that? That's a bird that finally completely died out in 1914. That's when the last individual died. But it had been under collapse for a couple of decades before that. What's so interesting in this case is this species probably had the largest biomass of any bird species ever. There were billions and billions of them. And in their migrations from western part of the continent over to the northeast, they would descend in huge numbers on trees where they fed. And that provided an opportunity for, guess what, market hunters to go out and blow away as many as they could. Passenger pigeon was on Delmonico's restaurant menu for many years. Because we could do it, we did it. Excuse me, which is so typical of us. Now, where are we with this particular project? We have the genome of passenger pigeons reconstituted from ancient DNA. We have the genome of its close living relative known as the band-tailed pigeon. And the effort in this case is to produce a hybrid in just the ways that we've been talking about. And by back crossing, eventually get something that is a true breeding lineage. Advantage? Well, if you remember my first reason for entertaining de-extinction, it's because we did the harm. This is restorative justice in action, to bring back a species that we were personally responsible for killing off. Now, what I like to say is in New York City, the last thing any of us are interested in is another pigeon. Fine. It's very unlikely that passenger pigeons are going to be able to recover to the numbers they had in the past, in part because the ecosystem is so changed. We have changed it. The kinds of places that they roosted, the kinds of foods they ate, all of that kind of thing is in much shorter supply than it ever was in the past. So do I endorse it? I endorse the effort because, again, to go back to my original list, we're going to learn a lot from the effort. Do I think it stands much chance of actuation? No.
Do you know anyone doing this kind of research related to insects and especially pollinators that maybe haven't gone extinct yet, but due to the types of pesticides and other things that we're using in the environment are getting close to that point?
Yes, but in this case, it would be much more like what we do with our food crops. In other words, we're not going into the full operation of cloning, or certainly not this level of genomic engineering. What we want to do is to introduce single genes that are able to do a job that we want. Reduce infestation in the case of food crops, for example. This is done all the time now by Monsanto and others. It's creating genetically modified organisms, which is another topic all by itself. We condone it, we endorse it largely in this country because of the benefit that accrues as a result of doing it. You have stable crops. You don't have enormous losses due to pests, due to disease, due to other kinds of problems, wilting. You get cultivars that are able to withstand the kinds of climate change that we're now experiencing, where things are getting hotter and hotter and perhaps drier and drier during the summer season. It's an arms race between those that want to infect or destroy and what we want to preserve for our own purposes. And for that reason, this kind of research is extremely important. I have no issue with it. Invertebrates would be the same kind of thing. Now, there are some examples. The monarch butterfly comes to mind. There's some other butterflies that are felt to be in very treacherous circumstances. But once again, you're dealing with a different kind of organism. And what's characteristic of most invertebrates is the production of enormous numbers of eggs, very few of which, under normal circumstances, are going to survive. So here, the effort would be better directed toward, again, introducing single genes for a given purpose. If there's a threat to that particular species, you can identify the threat. You know from another species there's a gene that might help here. Then you experiment and you see whether or not it would do any good. If it does do good, then you introduce it into the germline of respective parents of more and more eggs. Then you can do a very valuable thing in ensuring, once again, that a critically endangered species manages to survive thanks to human ingenuity. Thank you for that question as well.
So we have time for two more, and I want to ask this one that someone has submitted that kind of relates to, I think, probably a couple of the questions you have there on your screen. This person is asking if using these types of genetic engineering methods, would we be able to create something like a Pegasus, such as like a winged horse, or other kind of novelty animals that might be sold in part or only to rich customers, which kind of, I think, ties into this health consequences for the animal ethical concern. Maybe should this be banned altogether concern? So do you have any thoughts?
I see I'm talking to the jackalope constituency now. Well, in principle, you can do practically anything. Are we talking about Pegasus or are we talking about unicorns? I think I'm going to talk about the unicorn for a minute.
You can pick whichever you like.
So a unicorn was not a horse. That may come as a surprise to those of you who love My Pony. But if you take a look at its feet, it has cloven hooves, like a cow or a sheep or something like that. It was a mythical creature. But what's its big deal? Well, its big deal is that lovely horn at the front of its head. It turns out that a number of now extinct species in the larger group that horses are part of, called Perissodactyla, developed those kinds of ornaments, horns, at the front of their heads. In fact, living rhinos, which are also perissodactyls, do that today. So here I'm flying blind, but let's go for it. Practice, see if you can get, God knows, a horse to grow a lump at the front of its head and call that a unicorn horn. I don't really endorse it. I mean, I think that's obvious.
Right.
But it's not out of the question. This is genomic engineering that's a decade old. The capacity to use something like CRISPR, for example, that work was developed in 2015. It's that recent. And I might add the people who did develop it, two women scientists, Etienne Charpentier and Jennifer Doudna from Berkeley, received the Nobel Prize in chemistry for their work. That's how important it was seen for the future of this particular science. I want to end with one other thing when it comes to manipulation, as we do when we're breeding animals. You've got the equivalent of vastly changed organisms compared to their wild types right now. You've got those among your companion animals. French bulldogs are a good example. For reasons of apparent cuteness, for aesthetics, whatever it is, people like how they look. But how they look is the result of disruption of normal development of craniofacial features. So they have this terribly abbreviated snout, massive underbite, all kinds of associated respiratory problems because of other developmental pathways that were affected by choosing animals that had the phenotype, but there was less worry about all the other consequences. Now, if there's bulldog owners out there, don't throw bric-a-brats. I'm not singling you out. It's what we do all the time. This is humans in action. We want something, we go out and take it. If it's not exactly what we want, we change it and make it our own. in many cases not able thereafter to live apart from us. That's who we are. My only hope is that going forward, there will be equal respect given to maintaining what's left out there, just because for the same reason we can do it. We can make it happen so that we're less exploitative. So we strip fewer assets. So we destroy less of the planet just because we can. How's that for a sermon on the Mount?
It's excellent. And it really ties into a lot of the conversation we've been seeing on social media from people too about whether or not this is something that we should be doing just because we can, and how we've been affecting the environment. And maybe we should just stop doing that rather than trying to
just stop
things.
Nancy Reagan just said it. She was talking about something else, of course, but you take the point.
So we'll end with this final question.
Okay.
And it's very similar to one that came in last week too. So this is obviously our Darwin Conversation series. So
Good.
what do you think Darwin would think about de-extinction?
He would be utterly enthralled. He would be all over this. Now, in fact, in Origin of Species and his other books, Darwin was not terribly concerned with extinction. It happened, right? Things happen. He was much more interested in the origin of species, not in their end days. But this kind of ties the bow, when you think about it, around evolutionary biology. We have learned so much about how evolution works. We still know very little about how extinction works. Why do some species go down at a particular time under very often unknown circumstances? And why do others, not substantially different perhaps, manage to forge on through? Is it about them? Is there some critical defect in them that makes it necessary that they eventually disappear? Is it the environment that works on them? Is it a combination? Or all of the above? Darwin, just because of his interest in everything in the natural world, how it works, why it works, would have thought de-extinction represented a marvelous opportunity. Once again, I don't know, speaking for him, I guess I can say this, but I don't know this. He would be less attracted to the idea of bringing back the kinds of species that would have the hardest time fitting into the world as it now is, and would favor the idea, I think, of going after those kinds of species that would not only fit in perhaps easily, but once again, would confer some kind of tangible benefit that people could accept as being, I don't know, the price of admission back into a fauna. And I think that's okay. I don't think that's a bad thing. But overall, what I want to end with is just the idea that we're here now talking about something called de-extinction in ways that would not have arisen 20 or 30 years ago. There was no science for it to support talking about bringing back the disappeared. There was no way to do it. So it was just fantasy. Now we're in some borderland between a reality where it hasn't happened or has not yet been successful at least, and what it might be in future. Thinking again of those mammoths at the foot of Mount Rainier, walking across the landscape in 2050. I'm not going to be here to see that, but I hope some of you will if it comes to pass. Thank you very much. And Lauren, thank you
so much. It was a pleasure having you here with us this evening to speak for our series and this conversation that I've been able to have with you at the end. So applause, applause. I'm sure everyone out there is also applauding, and there's been some thanks coming in the chat as well. So to everyone who tuned in, thank you so much for joining us tonight. I did drop the link in the chat to make a donation if you are so inclined, if you've enjoyed our Darwin Conversation series. And if you're listening on Facebook, that's giving.uoregon.edu/mnch gift. And we hope to see you at our next program.
Lauren,
Thanks
I
for
just want
coming.
to say, a quick thanks to the audience as well. These Zoominars are incredibly weird and often hard to take for long periods. So I'm hoping for those of you who are able to last to the end that you enjoyed it. And I hope you think about some of these issues that we've raised tonight, both by me talking and your questions, because they're really, really important. So thanks and bye.
Perfect. Thank you.